Hi everyone, welcome to ResearchMD.com. My name is Pramil, Cheriyath. I'm a physician working in the United States. I work as a program director, internal medicine transitional residency in the United States, also director of research and assistant professor of medicine, two large medical schools. So we've been doing a series of lectures. We started on endocrinology, and yesterday we talked about oxytocin. Today another great topic, and the topic is antidiuretic hormone, ADH. What are the other names when you talk about somebody? Tell me, what are the other names of ADH?
Vasopressin.
Vasopressin, okay. So what is the other name? There's three names actually.
Arginine vasopressin.
Arginine vasopressin. So what do you see over here? You got the 9 amino acid. It's a peptide hormone. 8th position is arginine. That's what they call arginine vasopressin. Remember that. Three names, very, very important. The first one is like ADH. The second one is vasopressin. And the other one, 8th position arginine. Arginine vasopressin or AVP. Okay? First, what is this thing? We have to look at the structure. What is the structure up here? 9 amino acids. Somebody count. Cysteine, tyrosine, phenylalanine, glutamine, asparagine, cysteine, proline, arginine, glycine. You got 2 cysteine and disulfide bridge, my friends. Okay? And then this is the 9 amino acid. This is the structure of vasopressin or the ADH. Everybody got that? And it's produced in the hypothalamus and kind of transported into it's a neural hormone, okay? Because it's going to go through the neural channel and go into the posterior pituitary, release from the nerve endings from there, okay? So that's what it is. So let's look at the synthesis of ADH. How does it synthesize? Magnocellular neurosecretory cell bodies in the supraoptic and the paraventricular nuclei, right here, supraoptic and paraventricular. Most of the action is happening where? Supraventricular. Supraoptic, okay? Not like supraventricular. So remember, supraoptic, most of the things happening there. And everything before they need a precursor, right? So what do we have is prepropresophycin, which contain 19 amino acid. So it contains AVP, then something called a neurophysin 2, and then you got a glycopeptide in there. That is the precursor. What is in neurophysin 1 is in the oxytocin. Remember that? Like we gave the presentation. So when you look back, oxytocin, there's, the precursor is combined with the neurophysin 1. Here, you got neurophysin 2, okay? Good point to remember. And then packaged into the endoplasmic reticulum and Golgi apparatus, secretory granules, and then they're stored in the Herring bodies, and then finally during the transport, it gets cleaved during the transport, and then AVP, arginine vasopressin, comes out, okay? Very, very simple. Now, let's look at the stimulus for the producing ADH. What are the stimulus? Increase AVP, okay? You got like increase osmotic. Mainly thing I worry about decrease volume, dehydration, decrease ECA volume. What happened? Angiotensin 2 is also released to increase the body water, to increase the blood pressure, okay? And then increase osmotic pressure. When you have decreased ECA volume, is osmotic pressure goes up. Pain, emotion, stress, and all these conditions can also make the increase AVP, okay? Increase ADH. And 2 drugs we may want to remember, clofibrate, which is used for the cholesterol, and carbamazepine also. Those 2 drugs can affect AVP. Just remember that. Sometimes they can link that question together. Now, what are the things kind of decrease AVP? If there's, if there's like increased volume, if the ECA volume goes up, what happened? Or osmotic pressure come down, and then remember the alcohol. That's why when you drink alcohol, you pee a lot, right? Because your AVP is decreased. Remember that. Now, next thing we have to know, there's 3 receptors that ADH works on. Okay, V1, V2, V3. Most important is the V2 receptor, which is located in the collecting duct of the kidneys. Okay, that's the most important. But I think it's important for us to cover all 3 receptors. We want to know how it works. We'll focus mainly on V2 receptors. Okay, everybody got that? Now, let's look at the first one, V1 receptors. Where are the location? Somebody read it to me.
Vascular smooth muscle and platelets.
Yeah, vascular smooth muscles and platelet. Okay, all of these things, like when we have V1 receptor, ADH come in, this G protein couple receptor we need to know. Okay, G protein couple receptor, and which is a GQ type, right? And then what happened? You got phospholipase C get activated, okay, and then you got ionosyl triphosphate, and then calcium is get from the endoplasmic reticulum is released, a lot of calcium, because vasoconstriction, glycogenolysis, platelet aggregation, and the ACTH release. You got one more mechanism up here happening. When this phosphor, you got, let's go back again. G-coupled receptor, one more action happening on this side, right side. You got G-coupled protein receptor, GQ type, activate phospholipase C, and then you got 1,2-diacyl glycerol get activated, protein kinase C activated, and then you got activated protein. What does it do? Help in the cell growth. Okay, that's one, the V1. Now let's go to what? V2. Okay, V2, the main thing. If you don't remember anything, you have to know everything about V2. That's where all these things happening, okay? So V2 receptors, first of all, where are they located?
Principal cells.
Principal cells in the collecting duct. Okay, that's very important to know the location first. And then you get activated again. What did we say? Like G protein coupled receptor. What type? GS type. Remember, this is GQ, my friend. This is GS type get activated. Once it get activated, adenyl cyclase activated, that convert ATP to CAMP. You have protein kinase A, and then there is a phosphorylation of aquaporin. Aquaporin are water channels. There's three types: V1, V2. I'm sorry, A1, A2, A3. Okay, 3 aquaporin channels are there in the kidney. The main thing is aquaporin 2. Aquaporin 4 is kind of located in the brain, and aquaporin kind of 5 is pretty much in the salivary gland. Okay, remember that. So we're going to talk about main thing over here, aquaporin 2 channel. Okay, aquaporin 1, 2, 3 located in the kidney. 4 in the brain, 5th in the salivary gland. Remember that. So what are we going to talk about? Mainly aquaporin 2 here. Okay. So aquaporin, these are water channel containing vesicles in the cytoplasm in the cell. When you get activated, when you get the phosphorylation, they relocated to the transluminal membrane, to the luminal membrane actually. Okay. When in the luminal membrane, what happens? The water start reabsorbing, all the water from the collecting duct reabsorbed, come here through this and go back to the blood, increase the volume, your blood pressure goes up, and the things work, right? Let's look at one more time, very, very important. V2 receptors, okay? They're located in the collecting duct, principal cells of the collecting duct. Activation, G protein coupled receptor, which is GS type actually, and then activate adenyl cyclase, and then ATP is converted to CAMP, and protein kinase A get activated. You got phosphorylation of this aquaporin 2 channels, my friends, okay? And then once you get added, relocation to the luminal membrane, boom, absorb all the water, gushing and coming in through right here, come back to the blood, increase volume in the blood, your blood pressure goes up. Okay, very, very important to know the aquaporin channels and the V2 receptor in this thing. Okay, now we said V1, V2, there was one more left, V3. Where are they located? They're found mainly in the pituitary gland. Again, GQ coupled to G protein receptor. Just GQ here, GS here. Remember that. GQ protein, and they increase the intracellular calcium. It helps in the ACTH release, and it also helps in the neurotransmitter mechanism also. So it's good to know all 3 mechanisms right here. So I'm going to take a step back and let's look at the whole picture. That's the most important thing, benefit of having a whiteboard right here. We talk about this 9 amino acid peptide hormone right here. Arginine at the 8th place. That's called arginine vasopressin. Or I think you can get confused about the names, but remember all the name: ADH, AVP, and the vasopressin. All this thing you have to know. Okay, so just come up here, and we know this magnocellular neurosecretory cell bodies and the supraoptic and the paraventricular nuclei get released. You got a precursor right here, it need to be cleaved, and AVP is finally released. Okay, we need to know that. And then it's good to know the stimulus which increases AVP and which decreases AVP. What do we see? Increase AVP in osmotic pressure. Remember, angiotensin II role is important. Remember the drug clofibrin and carbamazepine. Somebody can ask you questions on that. And decrease AVP, there is decreased osmotic pressure, increase ECF volume. The main thing is the volume increases. And always remember the alcohol, when you drink alcohol, there's decreased AVP, so water get lost, okay? Water will not get absorbed into the blood bag. Water will get off, that's why you pee a lot. Remember that. Okay, now, So when it comes all this, now we need to know why this stimulus comes in producing the posterior pituitary. You got what happens in the hypothalamus. How do they know if the blood pressure is down or the osmotic pressure? They got osmoreceptors. What is the normal osmotic pressure? Is 285. Body want to keep it exactly like it is. Okay, 285. Maybe like a 1%. I never seen anywhere else it wanted to keep exactly at 285. So they try to work on it. So now they have to, once they find out the osmotic pressure is like low, osmotic pressure kind of goes up. That's what the V1, V2, channels kind of activate. Mainly V2 is channel, but let's look about V1, what happens. You got glucose. G protein coupled receptor, and then phospholipase C, okay, and then inosyl triphosphate, and then calcium release, and you got vasoconstriction, glycogenolysis, and platelet aggregation, and you got the ACTH release, okay, and then don't forget the protein activate protein on the other side. But main thing, remember, don't forget the V2. I'm just going to go over like one more time. I know we've been kind of going over. Cannot forget the what happens in V2 and the aquaporin channel. The V2 receptor is collecting duct of the principal cells over here. G protein coupled receptor with GS pathway, and then activate adenine cyclase, ATP into CAMP, protein kinase A, phosphorylation as the aquaporin channels, what their water channels. What happened? Water get reabsorbed back into the system, and then go back to the blood, and then your blood pressure go up. Okay, very, very important. Then of course, finally you got the B3. We look at the protein, the pituitary gland, GQ coupled to G protein, and then you have increased intracellular calcium helping the ACTH release. Okay, so that's in a nutshell. Thank you so much for watching. We'll be back with another presentation. Please help us if you could subscribe to our channel. It would be great. Thank you again. Bye.